Our third presenter is Mudabir Abdullah from the Faculty of Arts and Science. Mudabir is pursuing a PhD in biology. His thesis title is Towards Humanizing the Proteasome Core in Yeast, and his 3-minute thesis title is Baker's Yeast, a distant cousin with human recycle bin. Good luck, Mudabir.
Most of you will agree with me that recycling is a pretty good thing, right? We collect our waste and other unnecessary things, toss them in a trash bin and send for recycling. What will surprise you to know that each and every cell in our body essentially do the same thing. Yes, human cells also have a garbage disposal and a recycling system that keeps them neat and clean. One of the cellular garbage disposal system is called a proteasome complex or simply a cellular recycle bin. We cannot imagine to live in a dirty house without having a proper garbage disposal system, right? Same is the true with the human cells. If they are not able to clear their trash or recycle them, they become sick, potentially leading to several diseases that include cancer like multiple myeloma, neurological disorders like Alzheimer's and Parkinson's disease. Given the importance of this recycle bin in human cells, we need a model to study its role in different diseases. In my PhD, I'm trying to build this human recycle bin in a simple baker's yeast. Yes, the same yeast we use in a bread and beer. Now the question is, why yeast? First thing, working with the human cells is costly, it's very difficult, and often we don't have that much tools available. Whereas working with yeast is easy, it's simple, it's cheap, and it may come as a surprise to you that though the yeast does not look like us, but it is our distant cousin. And we share several thousand genes with the yeast. And guess what? This garbage disposal system is also common between the yeast and humans. As a synthetic biologist, I challenge yeast to live with the human genes. And so far, I was able to make 50% of this recycle bin in the yeast. My engineering simply involves cutting the yeast gene and pasting the human one. Engineering the entire human recycle bin in the yeast will be a great milestone in the field of synthetic biology. I believe that my humanized strands will serve as a great model to study different human disease, and thousands of genes can be directly used on these strands to see whether a particular drug can restore the normal functioning of the recycle bin. Thank you.
Thank you very much, Mudabir. We're already halfway through the English presentations. Our fourth presenter is Trish Osler from the Faculty of Fine Arts. Trish is pursuing a PhD in art education. Her thesis title is Mapping Artistic Experience, Transdisciplinary Approaches to Understanding the Creative Process in Art Education. Her 3-minute thesis title is Bright Ideas, How Disruption Sparks Inspiration. Thank you, Trish. Good luck.
Where do bright ideas come from? And how? Inspiration. When you're solving a complex problem, multiple networks in your brain are activated, generating possibilities, some mundane,